The topic of this video is chromosomal rearrangements. The types of chromosomal rearrangements we'll be discussing are duplications, deletions, inversions, and translocations. Repairing of chromosomal breaks can cause these sorts of rearrangements. Rearrangements can be unbalanced, in which some loss or duplication of information occurs, or they can be balanced, where there's no loss or duplication of information. A duplication happens when part of the chromosome gets doubled. You can have tandem or dispersed duplications, and they can be in the same or reversed order of the original. Unbalanced duplications affect gene expression because you're changing the amount of information you have, or gene dosage. A deletion is a loss of a chromosome segment, which can affect several genes. Deletions can cause unbalanced gene expression in a region, and the position and size of the deletion has a big impact on phenotypic consequences. Haploinsufficiency is when an organism has lost one copy of a gene and is left with a single functional copy of that gene. The remaining functional copy of the gene is not sufficient enough to produce the needed gene product in order to have normal function. So a defective gene product results. In heterozygotes, if the dominant allele is deleted, the recessive allele would be exposed. So you'd get pseudodominance, which is when a recessive phenotype results in heterozygotes, although a dominant phenotype should have resulted. This can be due to a heterozygous organism having an intact recessive gene on one chromosome and a deletion of the dominant gene on the corresponding part of the homologous chromosome. An inversion is when a segment of a chromosome is inverted or flipped 180 degrees. The two types of inversions are paracentric and pericentric inversions. Paracentric inversions do not include the centromere, whereas pericentric inversions do include the centromere. Paracentric inversions form a dicentric bridge, resulting in large deletions. Pericentric inversions exchange information unevenly, which results in duplications and deletions in both chromatids. In individuals heterozygous for an inversion, if recombination occurs within the inversion, recombinant gametes will have unbalanced mutation, and this will most likely lead to a lethal mutation since recombination would result in large duplications or deletions. And this would result in inviable gametes. If you have a small inversion, there's a low chance of recombination in the inverted region. But with a large inversion, there's a higher chance of recombination inside the inversion. So since recombination would result in a large duplication or deletion, there'd most likely be more inviable gametes with the large inversions. A translocation is when there's movement of a part of one chromosome to a non-homologous chromosome. They can be balanced or unbalanced mutations, and large translocations can lead to inviability. Translocations, inversions, or duplications can cause large phenotypic changes, such as increased or decreased expression of a gene. They could move a segment near or away from regulatory sequences or heterochromatin, and both of these could reduce or enhance expression. These rearrangements can also lead to new linkage relationships or the formation of novel genes.